Hello everybody, my name is Brenda and I'll be going over transcription in eukaryotes. So right off the bat, one of the biggest differences is that in eukaryotes, this process takes place in the nucleus. So throughout post-transcription modifications in eukaryotes, in step 1, a gene composed of exons and introns is transcribed to the RNA by DNA polymerase. And so something to note here is that exons only appear to be present in eukaryotic cells. So the next step, SNRPs remove the intron-derived RNA and splice together exon-derived RNA into mRNA. In the third step, after further modification of mature mRNA, it travels to the cytoplasm for protein synthesis. And just a side note here, so conversion of RNA to proteins is actually called translation.